Alors, bonjour à tous, bienvenue à, je pense, à l'avant-dernière présentation de la série scientifique avant la pause de l'été. Welcome everyone to the before last presentation of the scientific series at St. Mary's Research Center before the summer break. Today's presenters are a team that I met when I was at Harvard last fall, and as soon as I learned about the research topic, I felt that it was very much aligned with the values of St. Mary's Research Center and St. Mary's Hospital, generally speaking, where there's been a century of leadership in women's health, obstetrical health, and with research now getting closer to normal people's important health issues that tend to be sometimes forgotten. Donc, les présentatrices aujourd'hui sont Dr. Rachel Pope and Dr. Christina Farr. Les biographies des deux présentatrices seront sur le site web, alors je vous encourage de consulter, c'est vraiment impressionnant. Dr. Pope is an obstetrician and gynecologist. She has a fellowship in global women's health on the repair and reconstruction of obstetric-related vesicovaginal and rectovaginal fistulas. She's lived many places, including Malawi and Southern Africa for 4 years, and served as the medical director of the Fistula Care Center and fellowship director for Global Women's Health at Baylor College of Medicine. She's currently chief of Female Sexual Health Division and part of the Gender Care Program in the Urology Institute at University Hospitals Cleveland Medical Center. That is a long title, Rachel. It's quite impressive. She also leads the Global Health Program in the Institute, where there's an international collaboration between urologists, gynecologists in the US and 3 centers in West and East Africa. Her clinical practice surrounds female sexual health, vaginal reconstruction, and her research interests are in health disparities and social determinants of health. Dr. Christina Farr is an assistant professor of pathology and a member of the Center for Global Health and Diseases at Case Western Reserve University. She has a Canadian connection. She's done her PhD at UBC and completed her postdoctoral fellowship at the University of Manitoba. Her research is focused on host microbe interactions that influence human urogenital health and disease. Current research projects in her lab focus on HIV, cervical cancer, infertility, and transgender health. So Rachel, Christina, the mic is yours. And just making sure you can hear us, you can see us?
Yes. Yeah, okay. And you can hear us as well?
Yes, we'll put our end on mute so that we can focus on what you're saying.
Okay, merci beaucoup, and that's the farthest my French extends, so I hope you're okay with my English. And please let me know if our connection is not good or if it breaks up at any point. We're in a clinic, so I can switch to another line if needed. So I'm grateful to be here. Thank you so much for that kind introduction. We're excited to talk about our joint projects. The two, namely, that we're going to go through are the DIVA study, the defining of the microbiome of the neovagina, and satisfunction. These are my disclosures and our funding. And I would be remiss if I didn't recognize my colleagues. This is not a project of just Christina and I here. We have a whole team. She has a whole lab she'll tell you about. And there's quite a few clinicians and community stakeholders who help us work with the community, make sure that our priorities are set by the community as well, and make sure that we can get from point A to point B, where we're involving both clinical, community, and lab work. There's so many individuals that, I just like to recognize them and thank them for their time. This was our team from 2 years ago during one of our research dissemination meetings. We've got sociologists, anthropologists, psychologists, urologists, gynecologists, etc. And it's really this interdisciplinary teamwork that I think has strengthened our research. So I want to give you a little bit of background. So vaginoplasties are done by 2 primary ways. The predominant method is to use penile skin, and it's called a penile inversion vaginoplasty. And the technique that we've developed at university hospitals is actually quite different, where we take the peritoneal flap from the inside of the abdominal wall, and we use that to line the vagina. It gives a completely different surface of the vaginal canal where it's more mucosal, it's a little bit more moist, and it looks more natural. And that's why many patients have opted for it and have come to our center for it. It presents a completely different microbial setting, and I wanted to give you the context so that you could see exactly what we're doing in these surgeries. Not to bore you, I know you're not all medical, but I'm going to try to show you this really fantastic presentation that one of our fellows put together to give you more context. And I think I'm going to have to share my screen here, hit the other side. So one moment while I do that. Okay, I hope you can now see the video. Yep. Okay.
We present our technique for robotic-assisted peritoneal pull-through for gender-affirming vaginoplasty. Port placement using the Da Vinci Xi robotic platform is shown here. Care must be taken to balance the ability to reach to the deep pelvis with the ability to begin the anterior peritoneal flap dissection as cephalad as possible. Prior to docking the robot, bilateral transversus abdominis blocks are performed. We begin by taking down any sigmoid adhesions to facilitate creation of the posterolateral flaps. The left posterolateral flap is marked out, taking note of the external iliac vessels and ureter. The flap must be long enough to reach the pelvic floor. A robust flap is then raised along the markings, taking care to cauterize any mesenteric arcade branches which may be encountered along the medial edge. The ureter may be encountered laterally. The pedicle supplying this flap is located at the inferolateral corner of the flap. A similar dissection is completed on the right side. The dissection is then carried forward through Denambier's fascia to the pelvic floor. Using feedback from the perineal surgeon, the robotic surgeon breaks through the pelvic floor to meet the perineal dissection. The canal is adequately widened to the level of the levator muscles and hemostasis is achieved. The two posterolateral flaps are sutured together in the midline using 3-0 V-lock suture. The flap is then passed to the perineal surgeon and will become the anterior lining of the neovaginal canal. The distal edge of the canal is fixed to the vaginal introitus using running 3-0 monocryl suture. The anterior abdominal wall is then measured to to raise a flap which is at least 11 centimeters wide. We start as cephalad as the robotic arms will allow and carry this dissection inferiorly, taking care to leave the inferior epigastric vessels with the rectus muscle anteriorly. The peritoneum is partially dissected off of the bladder until we meet the lateral edge of the prior posterolateral flap dissection. The flap is tagged and passed to the perineal surgeon. This flap will line the inferior and lateral walls of the canal.
So now we're looking from the patient's perineum. This is the urethra. Now we see where the vagina has come together.
The vaginal sidewalls are closed bilaterally with running 3-0 V-lock suture.
I want to just show you sort of the general idea of where this tissue is coming from, and why it's I think different to be studying it. So let me just now share back to the other spot, which is 1 second. Okay. So what we wanted to do, the two studies, the one first I'll talk about the status function. It's more of the qualitative study, but we wanted to find out after our patients had been living with this new vagina for several months to possibly several years, what's happening with their sexual function. So many of them want to have a vagina because they want to be sexually active. It helps them to feel aligned with their identity, and it helps to actually improve their quality of life and their mental health as well. And so part of that, we want to make sure that as we're innovating in surgery, that we're actually making things that are functional for people and are going to work long term and not create additional problems. So we looked through the literature, and most of our validated sexual functions surveys or questionnaires are really not appropriate for women with a neovagina. And so we had a process where we included medical students and residents to look through, do a systematic review to look at all female sexual function tools, identify what's appropriate and what's not. And then we did a qualitative study where we interviewed 16 trans women who had had surgery to find out what was most important to them. And we initiated a community advisory board. So this was really important because this community advisory board has been helpful from beginning to end with both studies to really make us trusted individuals in the community and to help us understand where there are gaps, where patients really want to have more information, and I think really has helped with our recruitment. We distributed this survey to 50 individuals. We interviewed 30 of them to confirm that the findings were correct and validated. And then we refined the survey even further by using our expert panel of surgeons, psychologists who you saw earlier, and then also our community advisory board. We then distributed the survey to 100 additional individuals, and we found basically that the survey had high fidelity. There was good inter-rater corroboration. We looked through, these are the results from the first steps to see that there's so many issues that just need to be incorporated for this special population. We needed to look through lubrication. We wanted to make sure people were not having unwanted lubrication where things were actually causing discharge or discomfort. We need to make sure that sensation was not just from numbness, that they were having positive sensation. And then we needed to look through body image as well. And so these are some of the themes of our questions. We broke them into 8 domains eventually. And I think that in the end, we came together with a really great finalized survey. And the point that I want to make is how important it was to have this community advisory board. I think they helped us to identify where questions didn't quite make sense, where things needed to be refined or clarified. And then that helped us to get such good results from the 100 individuals. So from that 100 individuals, we found the mean age was 39. It ranged from ages 20 to 72. Most of them in our population are white, non-Hispanic. If you look here, most of our participants have had some college or college degrees. Most of them are employed, although this is a high rate of unemployment compared to the average population of the US. Gender identity, so most did identify as trans women, and just a few identified as non-binary or other. We had a real mix here of sexual orientation, and so I think that's also important to know when we're talking about the neo-vagina, so that we know not everyone is planning to use the vagina for penetrative intercourse, and sometimes this changes, relationships change as well, and so that will lead to some of the factors that we'll talk about later. And then, of course, relationship status was quite mixed here as well. Like I mentioned, we had strong positive correlations observed between each individual domain and the total score. We're still going through this analysis now, but I did want to present to you sort of the basics that we had separated into domains of anatomy, arousal, orgasm, pain, and satisfaction, and found that we could break up these domains to create a score that a person could just use to assess and hone in on where the problems were, but then also look at a total score for sexual function and satisfaction. And then we did find that there were interdomain relationships confirmed, including correlations between arousal and orgasm and satisfaction, which made sense to us. These are the 8 domains, specifically self-image of the genitals, desire, arousal, lubrication, orgasm, satisfaction, pain, and anatomy. So you'll see how this all goes together for the anatomical outcomes. And this was a heat map that we created to look at these survey domains and look for those correlations. So that I just wanted to very briefly cover, because I think what you'll find more interesting is the DIVA study. That's really where our collaboration comes into play. But I mentioned the other one because the satisfaction study helped us to develop a cohort of individuals who knew that we were trusted people to come work with. They wanted to contribute to research. I don't think in my years of research I've ever had such a committed group of research participants. People show up for their follow-up. They want to see research in this area expand. And so with that, I'll pass on to my partner here, Dr. Farr.
And I think just to add, I think the recruitment into the study was the fastest recruitment I've ever seen in any other studies that I've worked on. So that's really a testament to the work that Rachel did with this population before we started enrolling. So I come to this, my background is primarily in the vaginal microenvironment in cisgender women. And that's how I sort of ended up working with Rachel initially before we started this project. And we know in cisgender women, the vaginal microbiome is important for their gynecologic health. So the optimal vaginal microbiome typically has low diversity and high levels of lactobacillus. And this leads to a low vaginal pH and an intact epithelial barrier. And we have a homeostatic balance between the cells and cytokines that are present in this state. When the lactobacillus is depleted and replaced with a diverse array of anaerobic bacteria, vaginal microbial dysbiosis can occur. And this can lead to disruption of the epithelial barrier, as well as the secretion of pro-inflammatory mediators like cytokines, the activation of the local immune cells, and then recruitment of additional immune cells into the vaginal space, leading to a highly inflamed environment. And in cisgender women, this is associated with clinical symptoms like discharge, pain, and odor. And it's also associated with a variety of negative reproductive health outcomes, including increased risk of STI and HIV acquisition, preterm birth, and cervical cancer. So there's been little work done investigating the neovaginal environment in trans women who've had vaginoplasty. But any of the work that has been done has really indicated the need to study further what this microenvironment is. So this is data from a survey that was conducted by Jessica Prodger's group at the University of Western Ontario, where they were asking trans women after vaginoplasty what their gynecologic symptoms and concerns were. And they found that many of the women were reporting bothersome symptoms like odor and discharge, as well as some concerns with pain and post-surgery complications. And in this survey, they also allowed the participants to do write-in answers, and I've highlighted a few of the ones that they reported in the paper here, where the participants are talking about things like vaginal discharge and odor that seems to come and go. So one participant reports that if they have discharge and odor, but when they have a shower, it goes away and then it comes back, that their symptoms seem to recur about once a month. That they find that their neovagina is similar to that of a cis vagina, and that it has its own flora environment, that douching, which trans women are frequently recommended to do, seems to disturb their vaginal homeostasis. And then one participant reported that they had symptoms of bacterial vaginosis for 6 months and was told that this was normal. And they couldn't find, or their healthcare providers had little information on how to maintain a healthy microbiome in the vagina. And they really had to rely on the internet and strategies for natal vaginal care to determine what to do to care for their neovagina. So when I was a postdoc at the University of Manitoba, I had the opportunity to be involved in a small research project where we had paired neovaginal and rectal samples from 9 transgender women. And after quality control measures, we had 5 neovaginal samples and 7 rectal samples available for our analysis. And these weren't necessarily paired between participants, just based on the quality control that we had done. And then we had vaginal samples from a separate cohort of 30 cisgender women that we used as a comparator. So shown here is an overview of the cohorts. And you can see the time since vaginoplasty was a median of about 10 years, although it did range from 1.5 years to almost 36 years for one participant. All of the participants had had a penile inversion vaginoplasty, with a few having a sigmoid colon extension performed. The rates of vaginal complaints were similar between the cis and trans women at about 40%. 60 to 70% of the trans women were using hormone therapy, and this was primarily estrogen. So shown here is our overview of the microbiome from the cisvaginal samples, neovaginal, and rectal samples. In figure A, this is a summary of the data for each sample type. And then in figure B, this is the microbiome of each sample that we had. So if you're not familiar, sorry about that. If you're not familiar with these type of plots, in figure B, each bar represents one participant, and the colors represent the composition of the microbiome from that sample. So in both A and B, it's clear for the cisvaginal samples that they're primarily dominated by species of Lactobacillus, and that's really what we expect based on previous work of cisvaginal samples. And although we see a little bit of Lactobacillus in some of the neovaginal samples, the composition of the neovaginal microbiome was completely different and primarily dominated by species of Porphyromonas, Peptostreptococcus, and Prevotella. And what was surprising to us when we did this study was that the neovaginal microbiome didn't look like the rectal microbiome. We had thought that just due to proximity of the sites, that the rectal microbiome was likely the source of the bugs that we were seeing in the neovagina, and that didn't seem to really match what our data was showing us. So the rectal microbiome and the neovaginal microbiome seem to be unique. And one of the things that we noted when we started to look into the literature for these bacteria that we were detecting in the neovagina was that a lot of them were considered to be skin or penile associated. And as all of these participants had penile inversion vaginoplasty, we assumed that the penile microbiome must be what was seeding the neovagina, but we don't have any data yet to support this. So when Dr. Pope and I met, we talked about different research areas of interest, and we were both interested in trans health, and that led us to setting up the DIVA study. So the goal of this study was to define the microbiome in a population of patients who have mostly had peritoneal pull-through vaginoplasty, to look at the incidence of HPV and other STIs, and to correlate our findings to the surgical outcomes. So we decided to divide this study into two aims. In our first aim, we were doing a cross-sectional recruitment where we were recruiting participants who were at least 3 months post gender-affirming vaginoplasty and collecting samples at this one time point. And we chose the 3-month time point because this is when the trans women are typically cleared for sexual activity, with the idea that most of the healing processes should have occurred to have that be less of a factor, have less of an impact on what we were observing. So at the study visit, the participants completed a survey, provided a variety of swabs, and underwent testing for gonorrhea, chlamydia, HIV, syphilis, and HPV. In our second aim for the study, we wanted to look at the colonization and stability of the neovaginal microenvironment in trans women who were scheduled to undergo gender-affirming vaginoplasty. So this is a longitudinal study where we were enrolling participants on the day of gender-affirming surgery so that we could get a penile swab prior to surgery. And then we would follow them over time after vaginoplasty to collect the neovaginal swabs, allowing us to do that pre-surgery versus post-surgery comparison to see if our hypothesis that the penile microbiome is seeding the neovagina is actually true. So before I talk about our data, I'd like to acknowledge some current and former members of my lab who were really instrumental in getting this project started and compiling some of the data I'm sharing today. So Taylor, Diatra, and Alejandra have done a lot of hard work on this study with me. So shown here is an overview of the cohort. The median age was 38 with a range from 24 to 68. And the median age at the time of vaginoplasty was about 37. And we did have a really wide range here from 23 to 66. The time since vaginoplasty was over a year. And again, our range was from that 3-month cutoff to almost 10 years. And the majority of the participants, almost 80%, had the peritoneal pull-through procedure, and the remaining had penile inversion. And about half of the participants had had additional surgery or revision procedures for both issues like vaginal stenosis or loss of depth, as well as for aesthetic reasons. Rates of self-report for vaginal symptoms were very high at 76%, and this includes things like discharge, pain, odor, bleeding, and itching. And pretty much everyone was using some type of hormone therapy. It was primarily estrogen therapy. Some were using both estrogen and progesterone. I believe one was using progesterone only, and then we have one participant who has no self-report or prescription for hormones in their file. And this is some data from the survey that we had administered to the participants. And we have a pretty extensive survey where we're trying to get an idea of different neovaginal practices and exposures to try to identify factors that may be influencing the vaginal microbiome of the participants. And we've divided the data here up by surgery type, as well as summarized it for the whole cohort. So the majority of the participants did report having some sort of receptive vaginal sexual activity in the past 30 days, with only about 20% reporting that this was receptive penile intercourse. Condom use was, or always using condoms, was reported by about a third of the cohort. And about a third of the cohort also was reporting that they currently have multiple sexual partners. And there was a real mix in the genders of their partners. So we had some participants whose partners had a penis, some whose partners had a vagina, and some who had both male and female partners. And then the majority of the participants were dilating. 60 to 75% were reporting that they were douching, and then almost everybody was using some type of lubricant. So we sequenced the neovaginal samples we collected using 16S rRNA sequencing, and the results of that is shown here. We detected over 100 different taxa in the neovaginas of the participants. And again, for these plots, each bar represents one participant sample, and the colors represent the composition of the neovaginal microbiome. And what we found, oh, and I should point out the legend is ordered by abundance. So the most abundant bacteria we detected were Prevotella, Peptinophilus, Porphyromonas, Streptococcus, Fusobacterium. And what we found was really that the neovaginal microbiome was highly diverse and contained a lot of species that we don't typically find in the natal vagina. So for those of you who don't study the natal vaginal microbiome, I've included an example of what that would look like here. So this top plot is the same neovaginal microbiome slide from bigger from the previous slide. And then this bottom is natal vaginal microbiome samples from a separate cohort we have here in Cleveland. And you can see that the majority of, about half of the natal vaginal samples are dominated by species of Lactobacillus. And although we do have about half of the participants having a more diverse vaginal microbiome, the species that are really detected in the natal vaginal microbiome and the neovaginal microbiome are very different. So Lactobacillus, which we would think of as the hallmark vaginal microbiome bacteria, we don't see it at all or at very low levels, less than the neovaginal samples. And when we were trying to look for factors that could be influencing the composition of the neovaginal microbiome, the first thing we did was compare the composition based on surgery type. And there were no taxa that were differentially abundant between participants who had peritoneal pull-through or penile inversion vaginoplasty. And shown here is diversity measure, and there's also no difference in the diversity of the neovaginal microbiome between the two surgery types. And although this may be underpowered because we do have so few participants who've had penile inversion, we're not clearly seeing a difference between the two surgery types at this time. We then were interested in if there was any differences in the microbial composition between those who were reporting having neovaginal symptoms. And we did this comparison looking at both each symptom individually, as well as just reporting yes or no that someone had at least one symptom. And we found 4 bacteria that were differentially abundant between those who did and did not report symptoms, although as you can see here, none of them passed adjustment for multiple comparisons. But we were very interested, particularly in the Fusobacterium that was increased in those who were reporting symptoms, because it's been associated with adverse outcomes in other studies, particularly those of cancer. So we were just interested to see that this was one of the species that was differentially abundant. And then we have conducted a few other analysis of these samples. So we took the neovaginal samples and we ran a 27-plex inflammatory cytokine Luminex array. As shown here are the results for the 24 cytokines that we had good coverage in our data. And we've overlaid the presence of any vaginal symptoms, discharge, and surgery type. And we found that the samples clustered significantly by the presence of having any vaginal symptoms, with a p-value of 0.05 for those who were reporting discharge, also clustering together. And then all of these plots over here are looking at some of the cytokines that were differentially abundant between those who did and did not report any symptom, with all of them being significantly increased in those who were reporting symptoms, suggesting that there's a highly inflamed environment in those who have report of a neovaginal symptom. And we've also done metabolomics analysis using a mass spec-based approach. So shown here are, we had about 70 metabolites we were able to confidently identify with level 2 certainty. And so these are shown here in a unsupervised hierarchical clustering heat map. And then we've overlaid again symptoms, discharge, and surgery type. And we found that the metabolites also are clustering by the presence of symptoms, and that there's a trend for clustering by the presence of discharge. And this again is suggesting that there's a different metabolite profile in those participants who are reporting symptoms. So a lot of this lab analysis is very preliminary, so we haven't yet had a chance to integrate our microbiome, cytokine, and metabolite data to really see if there's any specific taxa that may be contributing to this inflammatory environment or to this different metabolite profile that we're seeing. We also have mass spectrometry-based metaproteomics data that we're currently processing bioinformatically, so I don't have that data yet to show you today. And then, did you want to take over the last?
Yeah, so back to the clinical aspect. So sexually transmitted infections, of course, we're interested in, and this population in the literature has been shown to have higher levels of sexually transmitted infections, mostly due to the idea that they are a vulnerable population, oftentimes cannot find employment and may resort to sex work. However, I have not anecdotally found that in our population, and so that's why we wanted to make sure to examine for that with this study. So we found no new HIV diagnoses. We did have a couple of patients who had HIV at baseline. No new chlamydia or gonorrhea, no new syphilis diagnoses, but really of what I think is highly important is that we found 4 individuals of our initial cohort of about 40 with high-risk HPV. This is very much a quandary because there should not be too much of like squamous epithelium in the neovagina when we're using the peritoneum for both the anterior and posterior vagina. And it's a little bit suspicious that high-risk HPV may be forming or living in the peritoneal tissue. It's an area that there's really very little, if any, research at all. And finding this, we felt was, it's a pre-cancer, and so we want to be able to follow it. A few of those patients otherwise did come back for sort of long-term follow-up where we're doing similar colposcopy or vaginoscopy to look for any changes to the tissue, to look for anything that would look like a pre-malignancy. We've done a couple of biopsies that all have been negative so far, which is great. So we don't know yet what this represents, but we think that 4 out of 40 is quite significant, and we do want to follow them long term. We did also swab for any patient who had receptive anal intercourse. We did an anal HPV test, and we found 2 positive there. And we will be we have sent those patients to our colorectal colleague who does like dysplastic examination for those patients. So kind of interesting information, or at least we think it's really interesting. And we're trying to convince some journals to find it interesting as well. And also some grant funders to also agree with us that it's interesting so that we can follow those individuals who tested positive long term.
So in summary, for our lab-based analysis, we're seeing high rates of neovaginal symptoms and postoperative complications in the cohort. The vaginal microbiome is diverse, and it doesn't resemble what we would normally see in a natal vaginal microbiome. Neovaginal symptoms were associated with increased inflammation, a different metabolite profile, and potentially some changes in the microbiome. And then we detected HPV and had some abnormal cytology results in the neovagina, which we're clinically quite interested in following up on. And then I just want to acknowledge the members of my lab, my collaborators at Case Western, including the Bergener Lab, funding, and the study participants for making all of this work possible. And we're happy to take any questions that you have.